I'm going to be talking about CRISPR-Cas9. Here's our DNA strand. The blue is the enzyme Cas9 that actually cuts the DNA, and the orange is the guide RNA which finds the target sequence. Now the guide RNA will go towards the correct part of the DNA so the mutation can be created. When the gRNA takes the Cas9 into the DNA will unfold a little so it is now single-stranded in one part. Cas9 will then come around the DNA and the gRNA will bind to the DNA locus. Because Cas9 is an enzyme and a protein, it is able to cut the DNA. In blue, you can see its molecular pincers that will cut it. The result of this is that the DNA now has a double strand. Because the DNA now has a double strand break, which is an unnatural event, an alarm will go off in the cell and alert something to try and repair it. This process will cause non-homologous end joining. When the DNA is repaired, it will insert or delete some base pairs. For example, the top base pairs begin with the start codon and the rest encode the protein. The bottom pairs are after CRISPR-Cas9 went into effect and there was an insertion at the bottom base pair. The base pairs will now read and code differently. The insertion causes a frame shift mutation, and in this case, the mutation created a stop codon which tells the ribosome to stop translating. We now have a truncated protein. The top is the Huntington protein from the gene, and on the bottom is the short truncated Huntington protein, as a result of the CRISPR-Cas9. So the protein would no longer be functional, and CRISPR-Cas9 turns off the Huntington gene.